Okay, so those are all examples of microevolution by natural selection. Mm.
Sorry?
Hi.
So you need it more?
No, wish squeegee stuff.
For me? Okay,
Yes.
just fill up this.
Oh, okay. I thought it was a different thing. Thank you very much. Thanks. Thanks, Davis. Very interesting. The majority, or they have found, research has found, if antibiotic resistant bacteria are left for a number of generations without being exposed to antibiotics, they actually revert to the wild type. So apparently, it takes more energy to be antibiotic resistant than it does to not be antibiotic resistant. And so it seems that if they're not exposed to antibiotics for a number of generations, that the majority of antibiotic resistant bacteria will become resistant to antibiotics again. The problem is that it's very difficult to stop people from taking antibiotics. Okay, thank you. Thanks, Davis. All right, so you know how it works. So bacteria generally die with antibiotics. However, randomly, every now and again, a mutation will occur where a bacterium becomes resistant to antibiotics, and therefore, if antibiotics are applied, that bacterium will survive, and therefore it is fitter, and it will produce offspring that resist antibiotics because they will inherit that mutation, and antibiotics are useless for treating them. Okay. And the story about it being caused by people not finishing a course of antibiotics is a bit distorted. That's not what it is really. Because it doesn't matter how many antibiotics you took, this one would still survive. You finished that entire course, this one is still going to survive. It's a random mutation. Okay. It possibly is because of that thing that they might revert back to being non-resistant. Okay, so there's a YouTube video for you to watch. Good. Okay. Antibiotic resistance tests. I'm just showing you this just in case you don't know how these things work. Okay. So what they've got is they've got a petri dish that has got agar in it, a nutrient medium, and it's covered with bacteria. So now the bacteria are this cream film on the surface of the agar. So before they start anything on this, what they've done is they've got a petri dish with agar in it, and they've infected it with bacteria, and the bacteria is over the whole surface, and it's this, in this case, this cream-colored bacteria. Then what they do is they plop onto that thing a number of little discs of paper, filter paper, that has been soaked in various antibiotics. So here there would be 1, 2, 3, 4, 5, 6, 7 different kinds of antibiotics. And what you can see is all of them actually kill the bacteria to varying degrees. So this antibiotic is better than this one. And this one is the least effective. Because look, it has killed some of the bacteria, but the little clear circle around it in which it killed the bacteria is much smaller in this one than this one is. So this one was quite good. Okay. Right. Now the bacteria is antibiotic resistant to certain antibiotics. And to which ones is it resistant? It is resistant to this one, mildly resistant to this one, mildly resistant to that one. But it is still negatively affected by that one.
Although what's the antibiotic?
Filter paper. Filter paper. So a little bit of paper cut out of a piece of blotting paper. Okay. You know the stuff you use in the lab. And it's been soaked in an antibiotic. So what they're trying to do is do realize in order to fix a variable, they need to put the same volume and quantity and concentration of antibiotic onto all of those.
Yeah.
So they're comparing those 7 different, or however many there were, kinds of antibiotics and their effect.
Strange, yeah.
So what they do is they take a piece of a filter paper, a little round thing, that's the same size. And by doing that, they're able to control how much antibiotic they put of each one of them.
Okay.
So then they take it with a pair of forceps, and they dip it into antibiotic for, I don't know, fixed variable, 1 minute, whatever,
Mm.
and then they pop it on. So it should all of those should have the same quantity of antibiotic. The independent variable is the type of antibiotic. So each of them is a different antibiotic on
Yes.
that paper.
So, and then all the grey areas, the...
Where the bacteria has died. So bacteria has died here. So if I'm a lethal fist fighter, and you're a lethal fist fighter, and I say to you, hey, I'm better than you, and we're going to be standing in a crowd. Okay, so the crowd is like the bacteria,
Yeah.
and you've got 10 minutes, kill as many people as you can. Okay. If I have a bigger, clear area around me than you do, I'm a better fist fighter than you.
More effective.
Okay. So it's exactly the same here. So the antibiotic that is on the paper is the fist fighter. The clear area around it where all the bacteria died is how effective it is. The greater the diameter of the clear area, the more effective the antibiotic. And so this antibiotic is not as effective as this one.
Yeah.
Okay. This one is slightly effective because you can see a little grey area around it. That one's a little better, but this one was the best.
Yeah.
Then that, etc. Okay, William.
And why is the one yellow?
No, I don't know. I knew somebody was going to ask and I have no idea. Because the interesting thing is it's not the same one.
Yeah.
How would you know which antibiotic you'd put where?
Like in your mouth and
No, here, this thing.
What do you mean, like, if you knew which ones were more effective?
Yeah, so you come to the end, you've got a result, and you go, this one in the bottom right-hand corner, and then somebody comes along and cleans dusts underneath your dish and turns it around in the other direction. It's not in the bottom right-hand corner anymore. You don't know which antibiotic it was.
Stick a little toothpick with a flag.
You put?
A toothpick with a flag.
No, okay, so remember the Petri dishes you worked with last year? You pick them up, and you put a little A, little B, little C, D, E, F, G, and that's where you put the discs. Okay. Why don't you put it on the lid? Yeah, lid gets taken off and somebody looks and then they put it on a different way around and oh, your experiment's gone. Okay.
So that's why it's yellow. So they know.
No, I don't think so at all. Because I don't think that's
maybe
that
it
marked.
reacts to the bacterianuses in
No, so that marking is underneath the Petri dish. It's not in the agar, it's not in the, it shouldn't affect the thing in any way whatsoever. Okay, I really don't know. Okay, all right, so example 2 of natural selection. So there are beetles with genes for brown coloration, and they escaped. No, they escape predation and they survive to reproduce more frequently than beetles with green coloration. And so more of the brown beetles got into the next generation. So if you look at this here, for example, the predator eats the more easily seen beetles lacking the brown gene. No, that doesn't relate to this at all. I don't know how drunk I was, Gus. I'm sorry. That has nothing to do with this. Okay. Right. So, beetles that have brown coloration against a brown background are more camouflaged than beetles that have green coloration. And therefore the bird would see the green one and eat the green one. It's like the white mouse and the grey mouse.
How doesn't it relate to the picture? It's what it does.
Escape predation. I wasn't drunk. I'm drunk now. Okay. Right, you're happy. Macroevolution, formation of new species, you know how to test that. Okay. Same mechanisms, mutation, migration, genetic drift, natural selection, cause macroevolution. It tends to occur over a longer period of time, but not necessarily, and we'll give you some examples of that later on. So they can produce major evolutionary change, especially if given enough time, and it will be a new species. Especially if the mutations were for things called key innovations. Okay, so in evolutionary biology, a key innovation is a novel, in other words, an unusual phenotypic trait that allows subsequent radiation, I need to explain that here, and success of a taxonomic group. So taxonomic, you happy with this term?
No.
Just means classification. So in grade 8, remember, you learned taxonomy.
Yeah.
It just means classification. So there's like plants and animals and prokaryotes and blah blah. Okay, all that ghastly stuff that you.
Decades ago.
Yes. Okay. All right. So here, when I'm talking about radiation, I don't mean radiation. I mean from one or a few kinds of similar individuals, a whole lot of really different kinds of individuals arose. Okay, so that's what I mean by radiation. So, in other words, spreading out in terms of phenotype, but spreading out in terms of what they are like, and therefore what they're able to do and where they're able to live. So if you consider all the organisms that used to live in the sea, and then along came a fish that was able to walk onto land, then that was a key innovation, to develop limbs from fins. Because now, those things which all had to stay in the water can go and live on land. What's the benefit of that? The time that they get there, there are fewer predators. They're much more easy able to survive. That's terrible English, but you know what I mean. Okay. All right, so examples of key innovations were a bony skeleton, and these are just some examples. There are many examples. Having 4 limbs instead of fins, having wings. Wow, that was awesome. Because there were very few predators up there. Could get away from predators. And then walking on 2 limbs instead of 4 limbs. Yo, this thing was amazing. Oh, you could go to a tree and you could pick off a whole lot of fruit, and you could carry the fruit home and you could eat it where you were safe. Okay. If you walked on 4 limbs and here's a tree with fruit and you wanted to eat a whole lot, you could only eat what you could eat while you were there. And if you stayed around for any longer, the predators would come along because they knew that that tree was in fruit, and so they knew to come and visit it every day to kill what was eating there. Whereas if you were able to go carry a whole lot home and you're all nice and safe, then you've got such benefits.
So for radiation, so like that one organism with a specific trait then reproduces and its offspring have the trait
Over.
over time.
Okay. Can you wait until we get to Darwin's finches and just make sure that we understand the same thing? Okay. There's actually a picture which shows the radiation now, so I'll explain it to you. William.
So the radiation part means this macroevolution allows them to go to a different setting.
Yes, absolutely. Because now you've got something that can eat a different kind of food, for example, or can do something that the other ones couldn't do. And by being able to do that, they benefited by having a new niche that they could live in, and they had different kinds of food, extra food. Here we go. Okay, Michelle, this is the one. So, and I will get back to this. This is called adaptive radiation or divergent evolution. And this is all about Darwin's finches, but we'll talk about that in more detail later on. And it basically goes, there used to only be one kind of finch in South America. And what happened is that kind of finch, some individuals of that kind of finch flew across and were able to reach the Galapagos Islands, which are about 600 miles, about 1,000 blah blah kilometers off the mainland of South America, off Ecuador. And when they got there, the Galapagos Islands are, although they're all quite close together, some of them are north of the equator and some of them are south of the equator. And they have different currents. And they have totally different climatic conditions. So you can go from one island, which is a desert. It's completely barren. It's got a few cactus growing on it. And you can travel in a boat about an hour. And you can get to a different island that has got different currents going past it, and therefore a totally different climate. And it's got a forest. It's got a rainforest. And it's got trees, and it's got lots of rainfall, and it's got grass, and it's got whatever. So what happened is, from that original ancestral finch,
You feel that?
Yeah.
Feel what?
It's like something
Very
stuck in the
shiny.
ground.
Sure.
I don't feel it.
Thought William was kicking actually.
Oh, I think maybe your bomb's not sensitive.
so from that original ancestral finch, that's the whole lot of kids. Finches went and lived on the different islands, and because there was variation between the individual finches, if a handful went over to that island where there were only prickly pear cactuses, and some of them had beaks that enabled them to eat the fruit of the prickly pear, those survived, the rest of them died. Okay, those ones with those long pointy beaks that enabled them to eat the prickly pear, because they had those genes, all gave rise to offspring that were able to eat the prickly pear. But the other ones that went to that island where there were lots of trees that produced seeds, then the ones that had seed-eating beaks survived there, and the prickly pear-eating ones died. Okay, so what happened is, from one ancestral species, there was evolution to a number of different species, because they had variation and they were exposed to different conditions, and some of them were fitter in that condition than others, but others were fitter in that condition, and therefore those were the ones that survived for longer. They got more food, they were healthier, they were fitter, they lived longer, so they had more offspring, and therefore the following generations had those genes, not the genes for eating prickly pears, because those long pointy beaks don't help when you're trying to eat seeds.
Isn't part of the Galapagos Islands desert because of lava, and that's like volcanic eruptions, and then that rock hardened. So and that goes by like the primary succession it's only really still that primary succession of plants because of the.
It is to a certain extent by how old the island is,
Yeah.
because some of them are older than the other, because it is a hot spot.
Yeah.
And what happens is an island gets made because there's a volcanic eruption underneath the sea, and the lava goes, and then it solidifies, and now we've got an island.
Yeah.
But what happens is, and do you do geography? You must do.
No.
No. That island then moves. It moves in that direction. It moves like 10 centimeters a year. It is. It's hectic. And then the next one,
Yeah.
and here it comes. Now this one's older than this one, and that one's older than that one. And yes, in terms of succession, this is older.
Yeah.
But depending on where they are relative to that current, they've got completely different climates.
Okay. So even if it's, but surely the newer ones will have more desert-like conditions because they don't have as many plants.
That is part of it.
Okay.
But part of it is not to do with how new it is. Part of it, I mean, really, you can have two that are very close to each other, the same age, very different. Very, very different to each other. Okay, you happy with the radiation, what I meant by the radiation? Okay, no, because now I've lost it completely. Okay, you happy with that?
Thanks.
Okay.
So,
But we will get back to that. You are listening, so?
So radiation is basically one parent species, but then splits into various different species in different locations.
Yes. So in those locations, you're going to call it different ecological niches. And the radiation is called divergent evolution or adaptive radiation. And those two terms mean exactly the same thing. Okay. So we're talking about key innovations. So, no. Okay. All right. So this was the development of the evolution, macroevolution of different vertebrate groups. And you know that the different vertebrate groups are cartilaginous fish. They have a skeleton made of cartilage, for example, sharks and rays. Bony fish, they have a skeleton made of bone. Amphibians, reptiles, birds, and mammals. Okay, so those are the groups of vertebrates. Now this particular structure, and you need to listen carefully because this is important. This particular thing that we've got here is called a cladogram. So it's showing you relationships between organisms, and there is, can I explain it first before you write, guys? There's an imaginary timeline. Okay, there's an imaginary timeline. Okay. Right, so what it's saying is here's the ancestor that gave rise to all vertebrates. It's a common ancestor. And that first gave rise to cartilaginous fish. And then was the development of a key innovation, a bony skeleton. Okay, and bony fish were evolved at that point. The next key innovation was four limbs. So the first group that evolved then were amphibians. The next was an amniotic egg, remember we discussed that. And so reptiles and birds and mammals have got an amniotic egg. The next key innovation is hair and milk. Now, reptiles and birds don't have hair and don't have milk, and therefore they branched off over here. Okay, if I said to you which group evolved most recently, it would be mammals. Because if you look at the timeline, they were the last group that evolved. Okay. If I say to you which group is most closely related to reptiles, it's birds, because they diverged more recently than reptiles diverged from any other group of organisms. Okay. Yep.
So can you just define a cladogram again, please?
Okay, so, and just listen very carefully to what I'm saying to you. A purist will say to you that a cladogram has got a line at a 45-degree angle to the horizontal, okay, and off of that, at 90 degrees, come other lines. Okay. And a purist will say that as a cladogram. Other people will refer to phylogenetic trees as cladograms, and they're not. So if you look here, this is what is called a phylogenetic tree. Okay, and it shows the origin of life down there, and it shows all the groups of organisms, and it shows how they are related to each other. Some people will call that a cladogram. The purists will have a fit. Okay, because that is a cladogram. Okay. And at each branch, just before each branch, there is something which differentiates this group or all of these groups from all of those groups. So I say to you, if I say to you, name the organisms which have 4 limbs, that have 4 limbs, then you would say everything above 4 limbs. Amphibians, reptiles, birds, and mammals have got 4 limbs. But bony fish and cartilaginous fish don't. Does that make sense? So you're dividing the organisms into different groups based on features that are shared and features that are not shared. And you can do two people can do a cladogram with the same group of items, and they may divide them differently. So I could do a cladogram, for example, on a whole lot of, you could empty your pencil case and do a cladogram on the contents of your pencil case. It's basically just a system of classification. But in terms of evolution, it's classification according to time. Okay. If a line does not reach here, then that group of organisms is extinct. Okay, Bonga, wake up. If a line doesn't reach here, that group of organisms is extinct. So, according to my ghastly cladogram that I've drawn here, whatever group of organisms this is became extinct at that time. Doesn't exist in the modern day. Yep. Who mad?
Me, I don't understand that.
The extinct thing.
Yeah.
So remember there's an imaginary timeline here. Okay? It goes from long ago to now, current day, current day, current day. And so because these groups all go up to the top, to the current day, there. Do you guys know this word extant? It's the opposite to extinct. Means still exists. But because this line didn't go that far to current day, it means it became extinct at whatever time that was. So that was 1920, and that group of organisms became extinct in 1920. Does it make sense? Okay. Do you need a 1-minute squirm break?
Yes, please.
Michelle, did you not feel the second
Like right,
The first one was like.
well, because right before she started climbing grounds, I felt one, and Michelle's head was like, oh. That's not what you felt, is it?
The first one was scary.
Quickly.
Yeah, I felt it in my chair, and the second one, like.
Oh, you didn't do this in the middle.
I realize now.
Oh, you're in trouble. You're in trouble.
What?
No, he didn't do that at the end of the first half an hour.
End of first and second half an hour. Creates off to 14th of July.